You're a black.
What's that?
Next.
You know, it's not that much further. And if you come in C,
So, what do you think?
it's a reasonable.
Oh, here come the new folks. Come on in.
There wasn't any last time in the report, so just
Okay.
or not.
I think it's okay. I mean, you should just. So I got this one. I should see. He wrote the
What's your name? Mark Johnson. Oh, no, no. I believe like about to me, because Mark was essentially retired and doing a little bit of consulting on the side. One fell swoop, I went early in September.
So you weren't going to take my chair? You were wondering if it was valid?
Well, I was just no. But you don't have to contact us. If you want to give
When I stepped out,
Yeah.
So, are you against
Okay. He should be. Oh, yeah. Like emeritus.
and you all probably know already that the purpose of this group is so that everybody who works on research, whether it be deans and chairmen or graduate students and postdocs, that everybody can enjoy the same science and enjoy discussing science with each other. And of course, bringing together folks from industry and academia. The website is freestanding, and to have videos as well as stills on this website. And the idea is that since a major purpose of this organization is close by here, went to grade school, high school, and he is, as I say, a very interesting family. He's one of triplets, which is interesting. And then finally he got into video and is now happily doing research on cell wall glycobiology of human parasites. Some of my favorite parasites, and that's no, you're thanking me for stopping. Yes.
Actually, the usual ones that time.
John Samuelson.
Yes.
So, First cousin of Barry Summers.
So while this is brewing up, I tried to tell David that what was important was that since I went to Boston University 8 years ago, I think I've sort of finished my graduate degree and my postdoc with the great Phil Robbins, who has forgotten more glycobiology, in particular biochemistry of glycan, amoeba, or Giardia is also the infectious form. And so it's worth knowing how the wall is made, each other goes from one eukaryote or another. They aren't reinvented very much of the time. In contrast, what is reinvented over and over again are lectins. And the wall of the amoeba essentially has a very limited number of proteins, one of which is extremely abundant. This is essentially a silver stain of a 2D gel to chitin, and that's the case for the chitinase, the isolated domain, J-lectin. So that would be the wattle, the wooden card, the lattice. And then the mixture, 2 dimensions, or in 3 dimensions, that's what I'm doing left to right. So this is a cut, this is a 3-dimensional picture. Essentially, it gets put on the surface kind of in little globs over time, and then it coalesces. This is the outer surface. The tennis balls here are the nuclei, each of the gluten, and it's in separate vesicles from the protein. So the sugar polymer and the protein are made in separate vesicles. They're made early on during encystation. Almost all of it is in the wall by a day and a half. The second player, this one I named after my daughter, comes in late. So it's just coming in a little bit when all the other stuff is there, and really doesn't get finished until about 72 hours. So you can't do that in the cytosol of bacteria because the cytosol is reducing, so it's only sulfhydryl. So you're not going to get these linkages, and you're not going to get the stability in the glycoproteins, Jacob glycoproteins perhaps, on the surface of the parasite. We call this the foundation phase. The wattle phase is when you have the chitin fibrils that are essentially being cross-linked by these multivalent lectins, slightly modified by the chitinase to that. I don't think anybody's ever identified anything for chitin as a starting point. I'll show you the structure of in just a second of the glucan synthase. And the thing that is weird about all of these enzymes that synthesize urea and they don't grow very well or very fast. It's actually frightening how fast a commercial chicken grows. I think you can practically look at it and see organisms that have very complicated life cycles. This is relatively more complicated than Amoeba and Giardia, in that there is an equivalent of a cyst form, which is up here on the upper left, surprising in that toxoplasma and malaria all at one point in time swallowed a little green alga, so a eukaryotic cell, plant cell that was making. And then there's a macrophage lectin, seed-type lectin called dectin-1. So this is essentially fluorescence. As it turns out, autofluorescence is a property that's been seen before. It's mostly been seen in the ascus of yeast by a French dude who likes to sort these things. And there are not just dozens, but over 100 of these, glycohydrolase domains. It's not a dumb thing to do. It just isn't terribly informative. But anyway, the point is that easily genetically manipulatable toxoplasma infect mice very well, but they don't infect cats. There's epigenetic changes that have gone on in these organisms. The bacterium that causes gastritis that grows at pH 2, right? It's this crazy ass bacterium, okay? That causes gastritis. But normally, no bacteria, other bacteria live in the stomach, and it's even worse when you go down to the duodenum, where the pancreatic enzymes, invited Jerry Fink over here, he could give you a talk about why some beer is clear and some beer is murky, and it all has to do with the mannans on the surface. But the point is that the stomach is and the bile salts and the proteases are a bad thing, but each of these parasites is, and certainly in Giardia and
with some inhibitors.
Right.
So the binding will be much less probably, right? If
if they don't have carbohydrates.
Yeah.
No, it's not
it's
clear.
just protein.
I did not the phosphate be directly connected to the serine of the 3. And those guys are very likely to be, they're long sugar polymers and still are alive. And, so these are foolishly resistant to lots of bad things. The difference between them and the kinds of glycans that are present on the surface of fungi also protect the yeast. So if you mess those guys up, you can't really understand. And glycans are used for quality control of protein folding. They're used for...
And you're finding many different types of glycans, although a similar motif in terms of the dog and walk.
Yeah, right.
But
fibrin and lactin.
some of these are considered like kind of remnants of larger families and not related to.
Well, these guys aren't related to each other
at all.
At all. I mean, they're all
and they're
over
using different glycans.
And they're using different glycans. They're using different lectins. It's a little hard to know where the heck they got the chitin synthase gene and amoeba.
That's what I was trying to get at. Is how.
Yeah. What is common ancestry?
Yes.
I would say the common ancestry that and this is stupidly speculative, but as you get older, you sometimes become stupider. I think that the common eukaryotic ancestor, which in general is very hard to describe because things have bottlenecked, and it's much easier to describe the common ancestor of extant eukaryotes.
What were the circumstances for that?
I don't think they ever quite figured it out, except for the fact that it was in the water supply. So in order to get rid of these organisms from the water supply, you have to filter through sand. Most of the water supplies don't do that, I think including the water that comes from the Quabbin Reservoir that a lot of us drink in the Boston suburbs. They decided that it was too expensive to do that for whatever stupid manipulation you did, which is almost exactly the same as what everybody had been doing forever.
But that
Yeah, and also in a lot of civil wars and things like that. Yeah, but it can.
Thank you very much for this.
This is fun.
I mean,
So our next meeting will be in early December.